DNA replication is a semi-conservative process in which one molecule of DNA is unwound so that the two separate polynucleotide strands can act as templates for new polynucleotide strands to be bonded to them, forming two new DNA double helixes. So the first step is the enzyme helicase, which comes along and unwinds the two strands, breaking the hydrogen bonds between the two polynucleotide strands. So as it does this, unraveling the two strands, they are left to kind of come out behind, and they now have exposed bases. If we look at first this side, we're going to have the leading strand, new strand attached to this one, so also through complementary base pairing. So first of all, the enzyme RNA polymerase is going to add a short strand of RNA, which attaches by complementary base pairing, and this is going to act as a kind of primer so that another enzyme, DNA polymerase 3, can kind of attach to it, and this can then allow free building units called deoxynucleoside triphosphates, or DNTPs, to attach to the primer, and it's going to travel in the 5' to 3' direction and attach along this strand to form a new double helix. So thanks to this enzyme here, it'll fall along so that this new strand can bind onto it. On the other side here, we're going to add the lagging strand. So the beginning is a bit similar. The same enzyme, RNA polymerase, adds the short strand of RNA, and the primer, DNA polymerase 3, is also then bound to it. And we can note here that this happens close to this area kind of here, which is called the replication fork. And so this is kind of where the two strands separate. Unlike for the leading strand, the DNTPs are going to be added on kind of to make short strands. So it's gonna, the enzyme kind of travels along, we're gonna have little short strands of RNA like this kind of, and then another enzyme called DNA polymerase 1 will come along and remove this enzyme here, and finally the enzyme DNA ligase will come along, and, well, not exactly like that, but it'll kind of join up the spaces in between the small strands to form the one long strand, so that we have our second molecule of DNA.